So, pancreatic cancer. What is pancreatic cancer? Pancreatic cancer is a disease in which cancerous cells form in the tissues of the pancreas. The pancreas is a gland located behind the stomach and in front of the spine, and produces digestive juices and hormones that regulate the blood sugar. 95% of cancers start in the cells producing digestive juices and are referred to as the pancreatic ductal adenocarcinomas, whereas the other 5% are accounted for by hormone-producing cells. These are referred to as pancreatic neuroendocrine tumors. Only 8% of pancreatic ductal adenocarcinoma patients survive after 5 years. They have a median survival time of between 6 and 10 months. But how is this related to my study? Well, the problem is that in 90% of cases during diagnosis, the tumour has already spread. That means that only 20% of those patients are qualified for surgery. Chemotherapy with a drug called gemcitabine is the preferred treatment administered after surgery, increasing survival time by approximately 5 weeks. Gemcitabine is a cell cycle inhibitor and therefore can stop replication of these cells. The issue is, recurrence rates remain significantly high and resistance to gemcitabine are its main clinical limitations. So what about the hypothesis? Well, how will I test this? Our hypothesis looked at cancerous cells which develop resistance and how cells will respond when combined with another drug inhibitor. And this one is ATR inhibitor. This is another inhibitor of cell cycle, but working via a different pathway to gemcitabine. The second aim was to see why it was developing resistance and how this enabled its survival. To test for this, we looked at a protein which plays a role in synthesizing DNA called ribonucleotide reductase. We used 2 different cell lines and measured the metabolic activity of one with a high and one with a low concentration of this protein. And then we hypothesized the low level led to resistance and enabled its survival. But what were our main findings of this study? Well, the first finding, we saw that combination of both drugs did play a larger effect in inhibition of replication of cancerous cells. However, the second finding showed the low levels of ribonucleotide reductase cannot be deemed responsible for the development of resistance and thus its survival when we administrate gemcitabine. So what about future research? Well, because of the success of using cell cycle inhibitors, looking at other proteins may also play a role in development of resistance. Repeating the results, well, the experiment was long-winded, and for this reason there was a lot of human errors to be made. The conclusion? Combination drug therapy was effective and is now in phase 2 of clinical trials, and hopefully we will begin to increase survival time after diagnosis. Low RO1 is unlikely to be responsible for resistance in gemcitabine.